Alright guys, video 1 for Sensation and Perception, Unit 5. This is one of my favorite units. It's a lot of fun. We get to do a lot of cool things. So let's jump right in. Today we're going to be talking about some basic principles about sensation, and then we're going to actually talk about vision as well in this video. So let's hop to it. Sensation is the process by which our sensory receptors in our nervous system and all those sensory neurons receive and represent stimulus energies from our environment. So, perception, on the other hand, is the process that our brains and our bodies go through when we organize and interpret this sensory information. And this enables us to recognize meaningful objects and events. Both of these are one continuous process. It's not like we have to cognitively say, okay, I'm going to touch this, and then I'm going to send this energy up my arm, this electrical current up my arm to my spinal cord. We don't have to think about it. It's not a cognitive process. It's not a stage-by-stage process. Sensation and perception, both of these things are continuous processes that our bodies go through. Bottom-up processing is analysis that begins with the sense receptors and works up to the brain's integration of sensory information. So that's easy to remember. Top-down processing, however, is information that's processed, is information processing guided by our experience and our expectations. So bottom-up processing, I'll show you. Bottom-up processing is most of the sensation and perception that we do on a day-to-day basis. Is, you touch something and it's hot. That's bottom-up processing. Your fingertips, your sensory neurons in your fingertips touch something, and then it shoots up your arm to your brain. Bottom up. Top down is when you already have an idea what's going on, and so your brain adapts and it guides this sensory experience by experience and expectations. This picture right here is really cool. I mean, see if you can count how many faces there are in it. This is your sensory and perceptual processes working together to help you sort out these complex images. So this is an example of top-down processing. Take a minute and read this paragraph. It says top-down processing. According to research at Cambridge University, it doesn't matter in what order the letters in a word are. The only important thing is that first and last letter be at the right place. The rest can be a total mess and you can still read it without a problem. This is because the human mind does not read every letter by itself, but the word as a whole. I think that's really cool, and that's a great example of top-down processing. Selective attention means that at any moment, awareness focuses only on a limited aspect of all that we experience. And the cocktail party effect is, it's referring to our ability to attend to only one voice among many. So when we're talking on the phone while driving, our selective attention shifts back and forth from the road to the phone. And this process of shifting attentional gears can entail a fatal delay in coping, actually. One analysis of phone records for the moments before a car crash found cell phone users were 4 times more at risk for a fatal car accident. Selective attention limits our perception as many stimuli will pass by unnoticed. So our brains do this selective attention because if we were processing all of the sensory information that's coming at us all the time, I mean, that would be overwhelming and we wouldn't be able to function. So selective attention is our brain's way of allowing us to block out certain things so we can focus on something and on the important things. Again, cell phone use in car accidents. Again, like I said, selective attention limits our perception as many stimuli will pass by unnoticed, and this lack of awareness is evident in studies of inattentional blindness. And this is failing to see visible objects when our attention is directed elsewhere. Hold on, I'm going to go back one slide and see if I can't get this video to pop back up. There we go. And we're going to talk a little bit more about this, but watch this video.
This is an awareness test. How many passes does the team in white make? Go! The answer is 13. But, did you see the moonwalking bear?
That's a really great example of unintentional blindness, something going on that you never see because you're not paying attention. Your attention is focused elsewhere. Another form of inattentional blindness is called change blindness, and this is failing to notice changes in the environment. There's also change deafness, choice blindness, and choice blindness. Choice blindness and choice blindness means that. Let me see if I can get you a good definition here. Change deafness occurs in one experiment. 40% of people focused on repeating a list of sometimes challenging words, and they failed to notice a change in the person speaking. The choice blindness was discovered in 2005, and it showed that what they did was show 120 volunteers 2 female faces for 2 to 5 more seconds, and then asked them which face was more attractive. They then put the photos face down and handed the viewers which one they had chosen, inviting them to explain their choice. But then sometimes with a little sleight of hand, the researchers would actually switch the photos and give them the photo that they didn't actually choose. And most of the people still, 84% of the people still explained why they chose it, even though they didn't choose it. And the pop-out phenomenon is simply what, like in this picture right here, this guy stands out because he's different from everybody else. This right here is an example of change blindness. This younger man is asking the older man for directions. A pair of construction workers rudely walk through the conversation, and now it's a completely different guy who's giving the directions, and the older man doesn't even notice. That's a good example of change blindness. Here's another good example.
Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
But how did you know?
Madam, as any horticulturist will tell you, one does not plant petunias until May is out. Take her away. It's just a matter of observation. The real question is, how observant were you? Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
This is actually a commercial for English TV, and it's a good example of, yes, we can definitely miss things that we're not looking for, and we do it constantly. So it's a really great video, and we have some more examples of this. When we study the relationship between physical energy and psychological experience, researchers in psychophysics, actually, which is the study of relationships between physical characteristics of stimuli, such as their intensity, and our psychological experience of them. They identified what we call an absolute threshold as the minimum stimulation needed to detect a particular stimulus 50% of the time. And this picture right here is a example of how many times we look at certain points in a picture. Basically, they monitored eye movement to see where is our eye drawn to and how long does it linger there. Again, an absolute threshold is the minimum stimulation necessary to detect a particular stimulus 50% of the time. The signal detection theory predicts how and when we detect the presence of a faint stimulus, assuming that our individual absolute thresholds vary with our experiences, expectations, motivation, and level of fatigue. And the signal detection theory, talking about the ratio of hits to false alarms, that's what the signal detection theory is. It measures how many times we actually see a stimulus or respond to a stimulus versus how many times we think we don't respond. I'm sorry, there was somebody at my door distracting me. The priming effect, as shown in experiments, reveals that we can process some information from stimuli too weak to recognize, and this indicates that much of our information processing occurs automatically and unconsciously. Remember priming from that video we talked about where we watched the amazing way our brains actually work? This is an example of a subliminal message right here. It says, love this text in the back. Subliminal messages actually don't work, but it's still, fun to do it. So subliminal means simply below the threshold. A difference threshold is the minimum difference between two stimuli that a person can detect 50% of the time. So in humans, difference thresholds experience like a just noticeable difference, that's the J&D, increase in proportion to the size of the stimulus. And this is known as Weber's law. Hold on, let me go back. Weber's law is the principle that to be perceived as different, two stimuli must differ by a constant percentage rather than a constant amount. And I'll show you an example of this. You don't notice that it's a just noticeable difference, and that the size of the text right here is much smaller than the size of the text right here, and actually the text is actually changed as well. Sensory adaptation refers to diminished sensitivity as a consequence of constant stimulation, and that's what sensory adaptation is. So constant unchanging images on the eye's inner surface fade and then reappear. And this phenomenon of sensory adaptation enables us to focus our attention on informative changes in our environment without being distracted by uninformative background stimulation. And what's happening in this picture right here is that a projector mounted on a contact lens makes the projected image move with the eye. So initially the person sees the stabilized image, but soon she sees fragments fading and reappearing. So that's what they do in these kind of studies. Although sensory adaptation reduces our sensitivity, it offers important benefits. So like I said, it gives us the freedom to focus on informative changes in our environment. Our sensory receptors are alert to novelty. If you bore them with repetition, and they free our attention for more important things, like stinky or heavily perfumed classmates don't notice their odor because, like you and me, they adapt to what's constant and detect only change. So that reinforces a fundamental lesson that we perceive the world not exactly as it is, but as it is useful for us to perceive it. So those are some basic ideas about sensation and perception, and now we're going to talk about vision. We all have the ability to convert one sort of energy to another, and a great example of this is our eyes. Our eyes receive light energy and then they transduce or transform it into neural messages that our brain then processes into what we consciously see. The energies we experience as visible light are a thin slice from the broad spectrum of electromagnetic energy. Our sensory experience of light is determined largely by the light energy's wavelength, and this determines the hue of the color, and it also determines its intensity. And the intensity influences brightness. So, this is a picture of the electromagnetic energy spectrum. The spectrum ranges from gamma rays as short as the diameter of an atom to radio waves over a mile long. And the narrow band of wavelengths that are visible to the human eye, those are actually enlarged right here, extends from the shorter waves of blue-violet light to the longer waves of red light. And the physical property of waves is shorter wavelength is high frequency, and those are blue. Long wavelength is low frequency, and those are red. Great amplitude is bright colors, and small amplitude are dull colors. So, light enters the eye through the pupil. And your cornea is this, the cornea isn't lit up right here, but the cornea is this area right out here, okay? After light enters the eye through the pupil, whose size is regulated by the iris, and I'm going to click on all these so you can get these definitions, especially with your vocab. But the iris is the ring of muscle tissue that forms the colored portion of your eye, and it controls the size of the pupil. So light goes through the cornea into the pupil. Again, the size is regulated by the iris, and a camera-like lens focuses these rays by changing the curvature of the rays, and this is a process called accommodation. And so the light, again, or the lens focuses the rays by changing the curvature and accommodating them, and they are then sent to the retina. And this is the light-sensitive inner surface of the eye containing the receptor rods and cones plus layers of neurons that process this information. So, cornea is the outer covering of the eye. The pupil is the adjustable opening in which center of the eye which light enters. The iris controls the pupil. So the iris is actually dilating and constricting in response to changing light intensity. A lot of times we say our pupils are dilated. That's not true. Your iris is actually dilating. Your lens is the transparent structure behind the pupil that changes shape to focus images on the retina. And then your retina receives these wavelengths full of rods and cones, processes this information. Rods and cones. Rods are the retinal receptors that detect black, white, and gray. Let's see if there's anything else you need to know. It's necessary for peripheral and twilight vision, and because cones don't respond to those type of things. And your cones are retinal receptors that are concentrated near the center of your retina, and they function in daylight or well-lit conditions. And the cones are really great at detecting fine detail and give rise to color sensations. Your retina's rods and cones are clustered around what we call the fovea. I'm trying to give you some time to get this all written down, or pause it or whatever if you want to. So, your rods and cones, again, right here, they're all clustered around what we call the fovea. Light enters the eye, and they transform light energy into what we call neural signals. And these signals activate the neighboring bipolar cells, which in turn activate the neighboring ganglion cells. Here's your bipolar cells. Here's your ganglion cells. And these axons converge to form the optic nerve. And here it is right here. And this carries information via the thalamus to the brain. And where the optic nerve leaves the eye, there are no receptor cells, and this creates a blind spot. The cones, which are located mostly in the fovea, enable vision of color and fine detail, and the rods enable black and white vision. Again, they remain sensitive in dim light, and they're necessary for peripheral vision. I'm clicking on all these so you can see them. I'm sorry. Again, this little image right here, you can do a little test. There are no receptor cells where the optic nerve leaves the eye, and this creates a blind spot in your vision. To demonstrate, close your left eye and look at the spot, and move, it says move the page, but you're going to have to kind of move your head to a distance, move the page to a distance from your face about a foot, at which the card disappears. And the blind spot does not normally impair your vision because your eyes are moving and because one eye catches what the other misses, actually. So here's your structure of the eye. And your fovea is the central focal point in the retina around the cone clusters. And your optic nerve carries the visual information through as neural impulses through the eye to the brain. So let's talk about visual processing. We process information at progressively more abstract levels, and the information from the retina's 130 million rods and cones is received and transmitted by the million or so ganglion cells whose axons actually make up that optic nerve. When individual ganglion cells register information in their region of the visual field, they send signals to the occipital lobe's visual cortex. And within this cortex, individual neurons, or feature detectors, and we're going to talk about feature detectors here, nerve cells in the brain that respond to specific features of stimulus like shape, angle, or movement. So once in the cortex, individual neurons or feature detectors respond to specific features of a visual stimulus. So the visual cortex then passes this information along to other areas of the cortex where teams of cells, called supercell clusters, respond to more complex patterns. Subdimensions of vision, color, movement, depth, and form are processed by neural teams working separately and simultaneously. And this illustrates our brain's capacity for what we call parallel processing. Other teams collaborate in integrating the results, comparing them with stored information, and enabling perceptions. This contrasts sharply with the step-by-step serial processing of most computers and conscious problem solvings. Some people who have lost part of their visual cortex experience what we call blind sight. And blind sight is a really incredible phenomenon. For example, when someone with blind sight is shown a series of sticks in the blind field, they report seeing nothing. Yet when they're asked to guess whether the sticks are vertical or horizontal, their visual intuition typically offers the correct response. And when they're told that they got them all right, they usually are astounded. So it seems that there's a second mind, or a parallel processing system that's operating unseen. So, visual information processing. Seen goes through the retinal processing, it goes through feature detection, parallel processing, and then recognition. And this all happens, as you know, as you're looking at this and reading this, it happens in split seconds. The Young-Helmholtz trichomatic, or the three-color theory, states that the retina has 3 types of color receptors, and each one is especially sensitive to red, green, or blue. So when we stimulate combinations of these cones, we see other colors actually. For example, when both red and green sensitive cones are stimulated, we see yellow. Let's see if there's anything else you need to know about that. It is important to know that most people with color deficient vision are not actually what we call colorblind. They simply lack functioning red or green sensitive cones, or sometimes both. And their vision, perhaps unknown to them because their lifelong vision seems normal to them, is what we call monochromatic, and that means one color, or dichromatic, which means 2 color, instead of trichromatic, like most human beings. And this makes it impossible to distinguish the red and green in this figure that you see in front of you. Dogs actually lack receptors for the wavelengths of red, giving them only limited dichromatic color vision. Herring was the psychologist that came up with the opponent processing theory. And it says that there are two additional color processes, one responsible for red versus green reception, and one responsible for yellow versus blue, plus a third black versus white process. So here's your three sets of colors. And so the opponent process theory says that in the retina and in the thalamus, some neurons are turned on necessarily by red, but turned off by green. And others are turned on by green, but off by red. And this opponent process theory helps explain what we call after images, and I'm going to give you an example of that. Stare at the center, just a second, yeah, stare at the center of this flag for 1 minute, then, like, you're going to have to pause this. So stare at it for 1 minute, and then click, press play, and let it go to the next screen. So go ahead and do that, and then press play again. After you tire your neural responses to black, green, and yellow, you should see their opponent colors here. So, or you could stare at a white wall and notice how the size of the flag grows with projection distance. So, I hope I didn't confuse you too much. We're going to do some activities in class with this, but that's the end of video 1. Thanks, guys.